Hi everyone, I'm Lanesa, and today I'll be talking about assessing active ingredients in skincare. So here's a brief rundown of today's presentation. We have two parts. In the first part, we're going to go through a brief overview of actives. We're going to talk about actives for different skin concerns and challenges to active delivery. In the second part of this presentation, we are going to focus on active evaluation and critically assessing the evidence behind different actives. This is extremely important whether you are a consumer looking to buy a product with an active ingredient and incorporate it into your skincare routine, or if you're a formulator that wants to incorporate a particular active into a product. To begin, what are actives? Actives are ingredients that impart some sort of benefit to the skin or help to address a skin concern. But before we dive in, it is important to make the distinction between drugs and cosmetics. Per the Code of Federal Regulation, a drug is used to diagnose, cure, treat, or prevent a disease. They are products that have been proven to affect the body in a clinically meaningful and relevant way, and they are subject to pre market approval. In order for a drug product to be marketed, they must either follow an existing over the counter monograph, which is a list of specific actives that can be used for certain indications, or they must go through the formal drug approval process with the FDA and file a new drug application. Cosmetics, on the other hand, cannot claim to treat or cure diseases. Any cosmetic that does is considered misbranded. So, cosmetics are not subject to the same approval process that drugs are and do not have to meet the same standards to prove that they work. Despite this, cosmetics can still contain active ingredients that impart some benefit to the skin. Actives help address skin concerns, whether that be dryness and hydration, hyperpigmentation, acne and scarring, aging, or redness. There are a number of well studied actives that have years of documented efficacy against different skin concerns. On this slide, we have a list of these well-studied actives. And for the sake of time, I'm not going to go through everything on this list, but I highlighted in blue a couple of active ingredients that are used across multiple concerns. Niacinamide, vitamin C, retinoids, and hydroxy acids are compounds that are active ingredients and have been powerhouses in the industry for years. A lot of them are multifunctional in the sense that they can impart multiple benefits to the skin. And if you were to search for these actives, they each have a decades long history of efficacy backed by data, numerous clinical studies, and well controlled clinical trials. There are other active ingredients that can be used to address these skincare concerns, but many of the newer actives are not as well researched and don't have the evidence to support their standalone efficacy in a product. When evaluating the evidence on actives, I find it helpful to assess ingredients based on a hierarchy. We want to look at the efficacy of these ingredients under real use conditions. And in doing so, we would like to make sure that the research that we are using to evaluate the efficacy of these actives are reflective of those real use conditions. So there are certain studies that may support the use of an active for a particular concern, but are not relevant to the topical application of that active. This is a funnel that goes from low evidence at the top to high evidence at the bottom. And it's reflective of the fact that there are tons of actives now, some that may have marginal efficacy data and are staking their claims off of low relevant studies, but there are only a few well researched and well understood actives. This brings us to an extremely important point. Having an active, even if it's well studied, does not guarantee that that active will work as intended. Earlier, we went through the difference between cosmetics and drugs and discussed how drugs have to show proof of efficacy and safety to be approved for sale. Cosmetics do not. There are biological and chemical factors that can influence the efficacy of a topically delivered active. There is a common misconception that it is easy for the skin to absorb substances that are applied to it, but this is not true. The skin is a natural barrier to penetration. Its function is to protect the body from the external environment, whether that be outside aggressors, stressors, or pathogens. It also functions to prevent dehydration and maintain our water content inside of the body. This barrier function also makes the skin an extremely challenging tissue to deliver actives to and through. An active compound that exerts its effect has to exert that effect through some target, whether that be a receptor in the skin or an enzyme or a particular cell. You have to get the right active to the right place in the right form at the right amount. And in order to do this, it's a lot easier said than done, but we have to find a way past the skin barrier into the viable layers of the epidermis. For example, say you have a molecule that acts on a receptor in melanocytes. Well, these would have to penetrate all the way to the basal layer of the epidermis because that is where melanocytes reside. In another example, you could have a UV active, and those form a film on the skin to protect against UV radiation. So, those are things that you don't want to penetrate, that you want to keep on the surface of the skin. Another active may need to reach the viable epidermis because it has to reach keratinocytes. The challenge is getting that active to the right place. And this is a challenge because most skincare actives are not favorable for delivery into the skin. There are many important characteristics that affect the deliverability of an active. So if you look over to the right of the screen, there's a schematic of the steps in active delivery into the skin. Now, we're not going to go through this whole thing because the entire schematic deals with transdermal delivery, but even in just looking at the top, this is what we're going to focus on in the cosmetic industry. Just getting an active from the product into the stratum corneum involves many steps. And then to go to the viable epidermis, they have to penetrate and diffuse into that layer, which represents a whole nother challenge. There are multiple different steps of that process that affect delivery and deliverability. Deliverability is the potential for a particular active to penetrate and diffuse through the skin. And there are certain chemical characteristics of these active ingredients that influence their deliverability. And these are size, hydrophobicity, ionization, solubility, and instability. I want to point out size because it's generally accepted that molecules over 500 Daltons are not deliverable through the skin. So when we think about large ingredients like hyaluronic acid or collagen, these are molecules that have molecular weights in the thousands, and they can provide surface hydration, but they're not going to penetrate the skin. They will just sit on the surface, despite what might be told to you. So this excerpt here at the bottom is a description of a hyaluronic acid serum from a brand. And look at some of the claims and think about the size of hyaluronic acid and how that would affect its ability to quote unquote sink in. Keeping this in mind, let's look at the supplier bulletin that I pulled from LinkedIn, because this talks about the absorption efficacy of collagen peptides with different molecular weights. I highlighted a couple things, but the first thing that I noticed, obviously, what we just talked about is that the size is over 500 Daltons. So this is probably too big to be delivered through the skin. And the next part, it says that they can enter systemic circulation in the body, and they also have this chart as evidence. And if we take a close look at this chart, it says absolutely nothing about absorption or systemic circulation. It's just a molecular weight characterization of the active. At the end, it says that collagen peptides can be well absorbed and utilized by the human body and skin tissues, and they can act precisely on the stratum corneum, epidermis, and dermis of the skin and supplement collagen by promoting collagen production and protein synthesis. The data that is presented here does not support these assertions or conclusions. And it is important to remember a salesperson's job is to sell you things, whether those things work or do not work, whether you need those things or do not need those things. If you're a consumer looking to buy a product from a company or a chemist looking to buy an active from a supplier, you have to be able to critically evaluate the evidence that is being put in front of you. That brings us to part two of our talk on active evaluation. And it is important that we never take what is said about an active at face value, whether that be from a company, a supplier, or from literature. Actives require special attention and evaluation. When we evaluate the evidence for an active, there's really three types of data that we'll be looking at. In the first column, we have clinical studies. And clinical studies are going to show the efficacy of an active ingredient. They'll talk about the dosage form, the concentration, the effects, and the adverse reactions. Suppliers and companies or brands, suppliers will have data on the ingredients that they sell, especially the actives, and they'll try to do some sort of testing to support the use of that active. However, you kind of like what we just looked at in the previous example, we need to take a critical look at the testing that they did to support uh, the claims that they're making about the active. And you need to just make sure that what they're saying matches up with the data that has been provided. The last type of evidence is going to be chemical characterization data. They're going to be characteristics of your compound. They're going to convey important information about molecular weight, hydrophobicity, solubility, ionization, and some characteristics of the active that will tell you about its stability and deliverability. One tool that I really want to leave you guys with is how to critically evaluate literature, how to go through some of the marketing spin and get to the real data and the real evidence. It's important to do these critical evaluations of the literature of any active because not every study is a good one. And you want to make sure that a study measures accurately and effectively what it wants to measure. It is a very common theme in the cosmetic industry to not have your study back up some of the assertions that you make. This is just something that we should all be aware of. Earlier, I also talked about how we want our studies to reflect real use conditions. Here's a list of things to keep in mind and considerations for when we are reviewing the literature. First, study design. What are the size of the studies? How long were they conducted? What type of study and what type of model that they use? These are extremely important to understanding the strengths and limitations of a particular work as it applies to topical delivery. For example, an in vitro study where cells were incubated with a particular compound is way different from real use conditions of topical application of that compound in a vehicle with excipients. Over on the method side, we want to critically assess some of what they did in especially like clinical trials. We want to look at inclusion and exclusion criteria. We want to look at the route of delivery. I cannot stress how important route is in looking at the safety and efficacy. You cannot take results from an oral evaluation and equate it to the topical administration of a product. You have to ask, are there adequate controls in this study? And the active that we're evaluating, is it being influenced by the presence of other actives in the formula? Was there adequate randomization between experimental groups? What type of measurements were we taking? Are they clinically relevant to evaluating the active? And how were these things analyzed? These are all extremely important to dissect when we're looking at the literature. Because everyone is looking for the next new active ingredient, the next niacinamide, the next vitamin C, the next retinol. When I say everybody is looking for this next new active, it's on the brand side and on the supplier side. Sometimes, and a lot of the time, outlandish claims can be made for certain new actives without the adequate evidence to back them up. Some quick trends that I have noticed lately. Um, Number one, derivatives. It is important to understand that derivatives are not the same as the original active, and changes in structure can influence delivery and efficacy. It doesn't guarantee that if you have a derivative of, say, retinol, like a retinol ester, that does not guarantee that it will have the same efficacy as retinol. Another popular thing right now are botanical extracts. We have to understand with these is that they're complex mixtures of ingredients. They have many components, and usually you'll get your active effect from a particular component of that mixture. But like all naturals, because they are natural products, they are subject to regional, seasonal, and batch to batch variations in their composition. Because they are complex mixtures of many different chemicals, they can cause irritation and sensitivity, and they're typically used in pretty low amounts. So you need to keep that in mind when we're thinking about the end concentration of the active in your formula. In line with the topic of new actives and critically evaluating new actives, on this page, we have an example of two clinical studies on tranexamic acid. And we're going to see that despite this having two clinical trials that look at the topical effects of tranexamic acid, it doesn't necessarily mean that they support the topical efficacy of tranexamic acid as a standalone ingredient. On the left, we have a double-blind randomized prospective study that looks at 5% tranexamic acid versus its vehicle. It was a split-phase trial and it lasted 12 weeks. They used multiple methods to evaluate pigmentation and erythema, which is good. You always want to see a study have multiple methods to assess the same thing. However, if you look at the results, it says that even though there was a significant reduction in pigmentation on both sides, there was no difference between the tranexamic acid gel and the vehicle. So even though there was a significant lightening effect achieved with the product, it did not outperform the vehicle. So the lightening effects that are observed are probably not solely due to tranexamic acid. On the right side, we have another clinical trial, and most notably from this one is that in the method, participants were instructed to not only apply a topical tranexamic acid ointment, but they were also taking an oral tranexamic acid supplement 3 times a day. So even though the study observed significant results in pigmentation lightening, there's no way to separate the effect of the topical from the oral because they were used together. So neither of these studies makes a particularly strong case for the standalone use of tranexamic acid in pigmentation products. Last but not least, you want to do a deep dive into the chemical characteristics of your active because understanding the chemistry of an active ingredient is important to its stability and to its efficacy. For example, a very popular active is vitamin C, and it's known to be highly unstable and prone to oxidation. So, when formulating with vitamin C, you're going to want to make sure that there are antioxidants and other excipients in that formula that are going to stabilize it and keep it in the correct form. These are things that you can learn from looking at the chemistry and the chemical stability of your active. So now you've done all this research to learn about your active and characterize your active. You've looked at the clinical trial data. You've pulled the suppliers' brochures. You've looked at the chemical characteristics and the stability to ensure that it's properly stabilized and in the right form. That means that you'll have a functional product, right? And the answer to that question is no. No, it does not. And this brings us to the importance of the individual formulation. As I said earlier, it's really challenging to do active delivery into the skin. And the overall formulation of your product matters, including the excipients and the other ingredients that are also present in that formula. Clinical results for an active are great, but just because a clinical trial observed a particular result with a particular active doesn't mean that your product will exhibit that same efficacy or even any efficacy at all. It's because we're not using the exact same formula that they were using in the clinical trial, and there are interactions with excipients within your formula that influence the delivery of your active. And that's why it is extremely important if you are formulating with actives to test your specific product both for its ability to deliver an active ingredient and for its efficacy. For consumers, this is important because it means that just because you buy a product with an active doesn't mean that that product is adequately verified to do what it says it's going to do. When you're evaluating products online with active ingredients, you still want to use an evidence-based approach to unravel some of the marketing spin. Every product has a marketing spin, and you want to try to work your way through the marketing spin to figure out what's really going to be effective. There's a difference between a brand saying something like, our product contains blank ingredient, which has been clinically shown to exert blank effect, versus a company or brand saying through clinical studies, our product was shown to do this. And I'm not going to get too deep into this, but just keep in mind the level of evidence that certain claims convey versus others. What I really want you to take away from this presentation is that although there are actives in skincare that can be beneficial to the skin, there are a lot of different parameters and factors that influence their efficacy and their deliverability. Having an active in a product does not mean that that product will work as intended. So it's important to do a robust evaluation of everything and all the information at your disposal about a particular product or an active ingredient. Doing this evaluation is going to help you cut through the fluff and either find or formulate products that are effective. That brings us to the end of our talk. I really want to thank everybody for listening. I hope that you enjoyed and learned something new. Hopefully, there are insights that you can take home, whether you're on the development or the consumer end of industry. Feel free to follow me on Instagram and TikTok, where I talk a lot about cosmetic science, or connect with me on LinkedIn. I'm going to answer any questions that you guys might have. And I also want to thank Jen so much for letting me give this presentation. And once again, thank you all for listening.